Hello everyone, my name is Nanfang Pan from West China Hospital, and my topic today is about the disrupted structural connectivity and gray matter covariance coupling and its associated transcripting profiles in ADHD. I have no financial interest to disclose. And my investigation is primarily about ADHD, which is neurodevelopmental disorders, as you may know, and characterized by inattentive, hyperactive, and impulsive phenotypes. As for its psychopathology, you may know that its disruptions in distributed brain structural networks in both gray matter and white matter has been widely investigated. And we primarily will use the following two approaches. In the DTI, the diffusion imaging, we may use the probabilistic tractography to establish the fiber connectivity networks, as we call FCN. And in the T1 weighted imaging, we may focus on the gray matter. And that's why we may use the probability density function to construct the morphological covariance network, as we call MCN. But however, the cross-model interpretation of structural connectance from the both gray matter and white matter has not been systematically investigated in ADHD. You might still be wondering why we want to explore their coupling patterns. And since the FCN may infer the white matter integrity and axonal trajectory, and the MCN is generated by estimating the interregional correlations in their microstructural properties. And the previous research may indicate that the morphological covariance is partially explained by the interplay between the mutually trophic effects mediated by the direct axonal connections. And however, the convergence between the FCN and MCN may also be attributed to the co-expression of some specific gene expression. So that's why which may need further investigations. And in this study, we have 23 ADHD and 83 ADHD and 89 healthy controls in the discovery sample, and we also have an independent validation sample with their neuroimaging and diffusion and T1 weighted MRI data. Then we use approaches of probabilistic tractography and probability density function of KL divergence similarity to construct the FCN and MCN metrics. Finally, the SC and GMC coupling features were quantified by computing the Spearman rank correlations between corresponding row of the FCN and MCN. And you may see the upper panel for the results of the coupling patterns. You may see, and we found that the stronger positive coupling features in the thalamus and the basal ganglia, while the negative coupling values was shown in the precuneus and the paracentral lobe in both groups. And based on those predefined network membership of brain pathos, we may find that there is a significant case control differences in the coupling values of ventral attention networks, as we also call it salience network, primarily includes anterior insula and the dorsal ACC. And then we employed the machine learning pipeline to identify ADHD from the children with healthy controls. We employed the nested cross-validation scheme within their inner layer to turn the hyperparameters and outer layers to evaluate the performance of the model to avoid further linkage of information and avoid the selection bias. And then we assist the unimodalities with assigned network-specific coupling features, and we only select those significant unimodalities in permutation testing to reduce the dimensionality. As for the classifier, we used the L2 regularized and L2 loose SVM and cross-validation ratio to evaluate the feature contribution. And then as a result, in the classification model, we established the multimodal fused classifier trained on the features from those significant unimodalities, especially models of VAN, DMN, and SMN. You can see the blue line of the ROC curve. And we achieved a balanced accuracy of 66.1%, and you may see the blue line, yeah. And when considering the external validation, the fused classifier could identify ADHD children in an independent sample with a balanced accuracy of 62.2%. And I think it means that our prior findings could be largely replicated. And then to determine those which coupling features would be relevant for the decision of the classifier, we calculate the cross-validation ratio and perform the sign-based consistency variable importance analysis. And as you can see in the left panel, we found that the coupling of the midline and the subcortical brain structures, especially the orbital frontal cortex, cingulate cortex, and hippocampus, thalamus significantly contributed to the classifier performance. And last, to bridge the gap between those identified neuromarkers and the transcriptomes, we extracted those open access gene expression data from the Allen Human Brain Atlas, which is the data coming from the six post-mortem brain, and to decode our findings, which contains information with more than 15,000 gene items, and we used the PLS regression analysis to select those candidate genes expression underlying those coupling features, followed by enrichment analysis to explore the gene oncology and biological processes. Then we explain the relationship between the case control differences in the coupling features and the gene expression patterns. You may see the upper panel, and this is the group differences for the whole coupling patterns, and the right panel is the weighted gene expression of the principal components. And we can find that the unturned coupling features and the first component of PRS score expressed and showed a spatial correlation, and the PRS1 significantly explained 12% of the variance of the case control differences in the couplings, which contains more than 300 selected genes. In the gene enrichment analysis, we found that the most relevant biological process of gene candidates is a neural projection development with gene counts more than 30. And the biggest limitation for my study may be that the machine learning predictive performance was not satisfactory enough. And there are some take-home messages for my study. We introduced a novel phenotype to delineate the brain structural connectome by quantifying the coupling between the brain green matter morphometrical similarity and white matter fiber connectivity profiles. And we established the corresponding classification model. And lastly, we explored their transcriptome correlates. And that's all I want to share today. I would like to express my gratitude to my supervisor, Qiyun Gong, and all other collaborators. And you are welcome to stand by to see my poster today. And thank you for listening.
Thank you for the great presentation. Can you give us some thoughts on, the validity of using something like the Allen Brain Atlas, which, where the participants have an age range that are, somewhat older than your ADHD subjects? How easy is it to interpret the findings when you do something like that? Between, comparing those subjects to a group of subjects that are very different developmentally.
And firstly, I want to clarify on the atlas that I use for this study. I use the Human Brain Net atlas to puzzle the brain regions, and which is a functional atlas for me to use. And maybe maybe the question you want to ask is about the neuropsychological meaning of these phenotypes. I think the larger the positive value of the coupling values, it means that the more green matter structural networks and white matter networks generated through these brain pathways tend to converge.
Yeah.
And the negative values indicate the positive side, and there may exist some group control differences here, and that's it. Thank you.
I have a quick question. Love the talk. Quick question on the use of a complex measure of brain anatomy. So do you have any thoughts about how to interpret the possible links between the gene expression relationships you're seeing and the complex structural phenotype? So how can we go about linking like a clear process to a complex brain phenotype?
Actually, my study is focused on the ADHD children, and the genetic data we extracted the open access data from the Allen Brain Atlas, which is derived from 6 post-term brain, which is from adults. And maybe it's a common way for us to investigate the brain and gene relationship, and I can recommend maybe some papers to introduce those metrics and methodology later. Thank you for this question.
Thank our speaker for a great talk.